Namaste to all. Welcome to the next session on molecular biology and RDNA technology. In this session, we are going to see about replication in prokaryotes. So in replication, DNA replication in prokaryotes, the important step in this is origin of replication fork. So in this session, we are going to discuss in detail about this particular part only. So if you see, we are going to take an example as Escherichia coli because in most of the process which has been discovered till now, it has been studied widely in Escherichia coli by most of the scientists. So as compared to eukaryotes, they don't processes or they don't possesses different stages. So it is a continuous process, that is immediately they undergo replication, then transcription and translation. So the mode of replication in prokaryotes, if you see, they have two types in this. The first type in this is called as theta model of replication, and second model is called as rolling circle model of replication. For understanding, we divide the process of replication as initiation, elongation, and termination. But as such, it is a continuous process, as we have said previously. So the bacterial cell division is taking place with the help of a process called as binary fission. So if you see the binary process of binary fission, we have already discussed in our previous session that binary fission is a process in which they form a groove or they separate themselves, and during this process only the DNA replication is initiated. So here what happens is, if you see, this is a bacterial cell wall, and then we are having the plasma membrane. Inside we have the DNA. So this DNA now it is present in the center or anywhere it will be present. But during the process of binary fission or cell division, the DNA get attached with the plasma membrane by the different processes which are there, like FTSZ, which is formed and it is attached on the top. So at that time only the replication transcription takes place. Once the replication is done, the other part actually moves to the other side and that also gets attached on the plasma membrane. And after this, what happens is the cell wall and the plasma membrane then begin to divide in between. So once they make the groove and the complete cell wall is formed between them, then what happens is they separate, and this process is called as bacterial cell division, that is binary fission. So you can see the imaging here. Here you can see the partially formed cell wall is there, and they are having the two nuclear materials on both the sides in the particular cells, that is Bacillus laciniformis, so where the division is taking place. So the theta model of replication, so if you see the theta model of replication, in this what is happening is, if you see, this is a circular DNA. So in the circular DNA, from the origin of replication, what near to the origin of replication, what happens is both the strand they make or opens up. Once it is opened up, it looks like a mathematical symbol theta. That is why this is called as theta model of replication. Once the newly synthesized strand starts to form and develop, the fork starts to form on both the sides and they elongate. Once they start to elongate, what happens is they reach to a particular area or point where now they have to separate. So after this, they break one of the strand of the DNA and again join back. By that time, the second strand which is there, they complete the process of replication, and other strand also completes or ligates and then completes the process of replication. So by this, we get the two daughter circular DNAs. And during the process of, as I said, during the process of binary fission, they get separated to different areas and the two cells are formed. Let's see about rolling circle model of replication. So in the rolling circle model of replication, one of the strand undergoes nick or a break. So what happens is, with the help of the DNA polymerases, the one strand where it has stopped, it starts to synthesize. And the other strand which has been formed as a nick, it separates and it actually forms or coils around. So it is like a rolling which is taking place here. You can see here how the rolling has occurred. So once the rolling has occurred, the other strand is also started synthesizing. Then what happens is the nick will be formed where the first strand will be separated and we will get double stranded DNA. The other strand which is formed here, that is the newly synthesized strand, which again undergoes the other strand formation and we will get double stranded DNA. So in this model, the rolling circle model, you can see that nick is formed, then it is getting it is being pulled out. So by that time it is pulling out, the other strand has started forming with the help of polymerase 3. And then what happens is it separates completely. By that time, it is one of the DNA is completely done. One of the daughter DNA is completely done. The other DNA then starts to synthesize the inner strand, and that also completes and forms as double-stranded DNA. So let's see about the formation of replication fork. So in the formation of the replication fork, we know that the DNA has double stranded, that is 5 prime to 3 prime and 3 prime to 5 prime. In prokaryotic DNA, they have ori site, which is origin of replication. In this ori site, what happens is the DNA melting takes place. DNA melting means the hydrogen bond which is present here, they start to divide. So due to the break in the hydrogen bonds, they start to form the bubble. Once the bubble is formed, then in this particular area, these are actually called as replication bubble formation. And with this, we have our fork, that is the origin of replication fork starts, where the newly synthesized strands starts to produce. So due to this, what happens is it looks like this mathematical symbol theta in the theta model of replication. So after this, process, we can see that on both the sides, it has the origin of replication fork. And in this replication fork, these are the replication fork which are being formed here. So after this, one of the strand is like anti-clockwise direction. It starts to move. In the other strand, it goes in the clockwise direction. So this replication fork, when we see here, one of the strand, it moves from this side and forms. So one of the strand, it completes like this. The other strand has completed in the same order like this. So it is a clockwise and it is anti-clockwise. Once it reaches to the point, the nick is formed and one of the strand is being then removed, and it forms as two separate daughter DNA, double-stranded DNA. So I hope that in this video you have got an idea how the different replication models are present in prokaryotes and how the origin of replication is formed during the process of replication. We will see the replication in prokaryotes in detail in the coming session. Thank you and have a good day.